In 2014, a team of researchers at the University of Pennsylvania published a genetic analysis that quietly buried one of the most widely held assumptions in modern science. The assumption was simple, almost intuitive, that the oldest human populations would be found wherever the oldest human fossils were found. Wherever bones were oldest, people were oldest. It seemed obvious. It was wrong. What the researchers found instead was that genetic age, the true measure of how long a population's lineage has been continuously unbroken, didn't match the fossil record the way anyone had predicted. The populations carrying the deepest, most ancient DNA signatures weren't the ones living near famous fossil sites in the Middle East or Europe. They were living in a corner of Africa that most of the scientific community had barely looked at. And when the broader scientific community began replicating those findings across dozens of studies through the following decade, using larger data sets and more powerful sequencing technologies, the conclusion kept getting stronger. There is one group of people on Earth whose DNA is so ancient, so genetically diverse, that their lineage represents the oldest continuously surviving human population that science has ever documented. Most people have never heard their name. To understand why this discovery was so significant, you need to understand what scientists actually mean when they rank populations by genetic age. They are not measuring how old individual people are, or even how old a civilization is. They are measuring something called nucleotide diversity, the density of genetic variation within a population's DNA. Every time a human cell divides, there's a small chance that a random, harmless mutation will occur in the copying process. Over thousands of generations, these tiny mutations accumulate. The longer a population has been living and reproducing in a region without being dramatically reduced by famine, disease, or forced migration, the more of these mutations stack up. Genetic diversity, in this sense, is a direct record of time. It is the closest thing biology has to a geological age measurement. Research published across multiple peer-reviewed journals, including work drawing from the Human Genome Project and data coordinated through the National Center for Biotechnology Information, established a consistent hierarchy. Non-African populations, those whose ancestors left the continent tens of thousands of years ago, show measurably lower genetic diversity than populations that remained in Africa. The reason is a mechanism known as a founder effect. When a small group separates from a larger population and migrates to a new region, they carry only a portion of the total genetic variation that existed in the original group. The farther they traveled, the smaller the founding group that established each new territory, and the lower the resulting genetic diversity. According to research synthesized through the Human Genome Project, this pattern plays out with remarkable consistency across the globe. The farther a population lives from sub-Saharan Africa, the lower its measured nucleotide diversity tends to be. Native American populations, whose ancestors crossed the Bering Land Bridge from northeastern Asia, somewhere between, in ways that are still clearly visible in genomic data today. East Asian populations show similarly constrained diversity, with the founding migrations into the Korean peninsula, the Japanese archipelago, and the plains of central China all tracing back to populations that had already experienced substantial diversity loss during the original out-of-Africa dispersal. European populations sit at roughly similar levels, their diversity shaped by a migration history that brought small groups of modern humans into contact with Neanderthals across the continent before the Ice Age populations contracted and then re-expanded. Researchers at the Max Planck Institute for Evolutionary Anthropology have confirmed that modern Europeans carry between 1% and 2% Neanderthal DNA, a trace of those ancient encounters, but their overall nucleotide diversity still reflects the bottleneck of their origins. None of this is a statement about intelligence, culture, or capability. It is purely a record of geography and time. But here is where the data starts to become genuinely astonishing. Because when researchers turned their analysis toward populations that had never left Africa, the diversity numbers didn't just rise modestly, they rose dramatically. And within Africa itself, the gradient continued. East African populations showed higher diversity than Middle Eastern or South Asian groups. West African populations showed even higher numbers. But sitting at the very top of every ranking, in study after study, was a group that many outside of genetic research circles had never encountered. Their diversity levels weren't just higher than European or East Asian populations, they were higher than almost every other population on Earth. And hidden inside that diversity were genetic lineages that diverged from the rest of humanity so long ago that some researchers initially struggled to believe the numbers. The Sand People of southern Africa, also called the Khoisan, did not make headlines the way ancient Egyptian discoveries do. They did not have pyramids or written records that drew archaeologists from around the world. For most of recorded Western history, they were either ignored or profoundly misunderstood. But when geneticists began sequencing their genomes in earnest during the late 2000s and into the 2010s, what emerged from the data was nothing short of extraordinary. A study published in Science in 2009, led by researchers including teams from the University of Pennsylvania and Stanford, sequenced the full genomes of 4 San individuals alongside other African populations. The findings confirmed what earlier mitochondrial studies had suggested but never fully proven. The San carry more genetic diversity within their population that exists between Europeans and East Asians combined. That single finding repositioned every conversation about human ancestry that had been happening in academic circles for the previous 30 years. To appreciate the weight of that conclusion, consider the numbers directly. When geneticists measure nucleotide diversity, they calculate the average number of genetic differences per base pair between 2 randomly selected individuals from the same population. For European populations, the figure sits at roughly 0.06 to 0.07%. For East Asian populations, the numbers are comparable, sometimes marginally lower depending on the specific subgroup being analyzed. For sub-Saharan African populations broadly, the figure climbs to approximately 0.1%. But for the San, specifically, the measurements push beyond that already elevated baseline. Because the human genome contains approximately 3 billion base pairs, a difference of even a fraction of a percent translates into millions of distinct genetic variants. 2 randomly selected San individuals will differ from each other genetically in ways that dwarf the differences between a person from Norway and a person from Japan. The San are not one branch on the human family tree. They represent multiple ancient branches that have been growing separately for longer than most of the rest of humanity's lineages have even existed. The explanation for this extraordinary diversity lies in what scientists call deep population structure. According to research published in the American Journal of Human Genetics and corroborated by work through the African Genome Variation Project, the San descend from populations that diverged from other human lineages somewhere between 100,000 and 150,000 years ago. That divergence predates the primary out-of-Africa migration. It predates the colonization of Europe and Asia. It predates most of the major events that shape the human populations living in other parts of the world today. While the ancestors of modern Europeans were tens of thousands of years away from even beginning their journey out of Africa, the ancestors of the San were already a genetically distinct population, accumulating mutations, adapting to the landscape of southern. The mitochondrial DNA record tells this story with particular clarity. Mitochondrial DNA is passed exclusively through the maternal line, making it an exceptionally clean tool for tracing ancestry across generations. The oldest known mitochondrial haplogroup, the genetic branch, designation researchers call L0, is found almost exclusively in San populations. A haplogroup in this context is essentially a clan marker, a genetic signature shared by all descendants of a particular ancestral female. According to research coordinated through the UMBER phylogenetic databases at the European Bioinformatics Institute, the L0 haplogroup split from all other human maternal lineages between 100,000 and 200,000 years ago. Every other mitochondrial haplogroup found anywhere else on Earth, whether in South America, East Asia, Northern Europe, or the Pacific Islands, descends from lineages that branched off after L0 was already established. The San. Maternal line is not just old, it is the root from which the rest of human maternal diversity ultimately grew. The paternal record, tracked through Y chromosome haplogroups, tells a parallel story. The deepest known Y chromosome lineage, designated haplogroup A or 0, was identified in a 2013 study published in the American Journal of Human Genetics. Researchers discovered it in a small population in Cameroon, and the estimated divergence date was so far back, between 200,000 and 340,000 years before present according to some calibrations, that it technically predates the anatomical emergence of Homo sapiens as a distinct species. While AO0 is specific to a Cameroonian population rather than the San directly, its existence reinforced what San genomic data had already suggested. African populations broadly, and Southern African populations particularly, carry Y chromosome lineages that stretch back into a period of human prehistory that most people's mental model of history doesn't even account for. But the San genome contained something even more unexpected, something that the original studies had not fully anticipated and that subsequent research has continued to investigate with growing fascination. Hidden inside their DNA were faint genetic echoes of populations that no longer exist. Ancient lineages that appear to have contributed ancestry to the San that don't match any known human group. The deeper researchers looked, the more complicated the picture became. In 2020, a study published in Nature Communications sent a quiet ripple through the paleoanthropology community that has not fully settled since. Researchers from the University of California, Los Angeles, working in fit neatly into any known human lineage. What they found was a genetic signal that could not be explained by any documented population. Somewhere between 250,000 and 300,000 years ago, the ancestors of the San had interbred with an archaic human group that has left no fossil record scientists have yet been able to directly connect to the genetic evidence. The researchers called them a ghost. Population. A group whose existence we can only infer from the DNA they left behind in people living today, because no bones, no tools, no archaeological site has yet been definitively linked to them. The concept of ghost populations is not new to genetics. Researchers have known since the early sequencing of Neanderthal DNA, published through the Max Planck Institute for Evolutionary Anthropology in 2010, that modern humans interbred with archaic relatives during their migrations out of Africa. The Denisovans, known only from a finger bone and a few teeth discovered in a Siberian cave, are another example of a group whose existence was confirmed entirely through genomic analysis before any substantial fossil evidence was identified. But the ghost population embedded in sand DNA is different in a critical way. It is older, significantly older, and it was encountered not during a migration into unfamiliar territory, but in Africa itself, in the region where modern humans were already emerging as a species. This means that the deep human prehistory of Africa was far more complex, far more populated with distinct human lineages than the textbooks written even 20 years ago acknowledged. According to modeling work published in PLOS Genetics, this unidentified archaic group may have diverged from the lineage leading to modern Homo sapiens somewhere around 700,000 years ago. That places them in a category of human relatives that is older than Neanderthals, older than Denisovans as currently understood, and representative of a branch of the human family tree that evolved entirely within Africa. The San carry approximately 2% of their genome from this source, a figure strikingly similar to the Neanderthal contribution in non-African populations, but pointing to an entirely different episode of prehistoric contact. What makes this particularly significant is that it demonstrates Africa was not a single unified cradle of one human lineage waiting to disperse. It was a continent of multiple evolving human populations, some of which merged, some of which disappeared, and some of which left traces that only became visible when modern genomic technology became powerful enough to detect them. The implications extend beyond the San specifically. Research led by geneticists at the Wellcome Sanger Institute and published in the journal Cell has found evidence of ancient admixture events in other African populations as well, including groups in West and Central, interacting across tens of thousands of years, and the populations living in Africa today are the inheritors of that entire complicated history in ways that populations outside Africa simply are not. The out-of-Africa migrants carried a subset of this diversity when they left. The populations that remained inherited all of it. This is where the ranking of populations by genetic age becomes most intellectually interesting and also most frequently misunderstood. When people encounter the finding that the San have the oldest DNA on Earth, the instinct is often to interpret that as meaning they are somehow more primitive, less evolved, or frozen in an earlier stage of human development. That interpretation is not just incorrect. It is the precise opposite of what the data shows. A population that has been continuously present in a region for 150,000 years has had 150,000 years of natural selection operating on it. The San have adapted to the arid landscapes of southern Africa with a sophistication that reflects that entire span of evolutionary time. Research published in Genome Biology and Evolution has identified unique genetic variants in San populations related to metabolism, sensory perception, and immune function that exist nowhere else in the human species. These are not primitive traits. They are highly refined adaptations that accumulated over a time frame that makes European evolutionary history look brief by comparison. The San also show evidence of genetic adaptations to diet that are unlike anything documented in other populations. Variants in genes related to fatty acid metabolism suggest long-term adaptation to a hunter-gatherer diet rich in specific animal fats and plant compounds native to southern Africa. Variants affecting bitter taste perception are more diverse in San populations than in any other group studied, likely reflecting tens of thousands of years of navigating a complex landscape of edible and toxic plants. Every layer of their genome tells the same story, not primitiveness, depth. In 2023, the H3 Africa Consortium, a pan-continental research initiative linking genetics laboratories across 15 African countries, released what is currently the most comprehensive genomic dataset ever assembled from African populations. The dataset included whole genome sequences from over 180 distinct ethnic groups, many of which had never been included in any prior large-scale genetic study. The findings confirmed and extended everything the previous decade of research had established. But they also revealed something that researchers had suspected but never been able to demonstrate at this scale. The genetic diversity present across African populations is so vast, so structurally deep, that current genomic reference databases used by the global medical community are fundamentally incomplete. The tools that hospitals and pharmaceutical companies use to interpret genetic data, to identify disease risk, to calibrate drug dosages, to understand inherited conditions, were built primarily from European genomic data. They are missing the majority of human genetic variation. And that gap has consequences that go far beyond academic interest. A study published in the New England Journal of Medicine examined the performance of standard polygenic risk scores, the genetic calculations used to predict a person's likelihood of developing conditions like heart disease, type 2 diabetes, and certain cancers across populations of different ancestries. The scores performed well for individuals of European descent. For individuals of African ancestry, the predictive accuracy dropped substantially. For individuals from populations like the San, whose genomic architecture diverges. Most dramatically from the European baseline, the tools were nearly useless. This is not a minor technical limitation. It means that the populations carrying the oldest, most medically valuable genetic diversity on Earth are simultaneously the populations least well served by modern genomic medicine. The scientific community is increasingly vocal about this problem. Researchers writing in Maasidai, Nature Medicine, have called it one of the most urgent equity failures in contemporary biomedical science. The practical response is already underway, though not yet at the scale the problem demands. The African Genome Variation Project, supported in part through the Wellcome Trust, has been systematically sequencing genomes from underrepresented African populations and building reference panels that can improve the accuracy of genetic analysis for people of African ancestry. Specific research programs focused on San and Khoisan genomics have been established in South Africa, with collaboration from institutions including the University of Cape Town and the South African Medical Research Council. These efforts are generating data that is expanding the known catalog of human genetic variants, including variants that affect drug metabolism, immune response, and neurological function in ways that have direct implications for global medicine. The oldest human DNA on Earth is not a historical artifact. It is an active scientific resource, but the significance of what the San genome represents reaches beyond medicine into something more fundamental about how our species understands itself. For most of recorded history, the narrative of human progress has been written by populations that are, in genetic terms, relatively young. The civilizations that built written languages, that constructed the institutions of science and philosophy that shaped the modern world, were largely the product of populations whose ancestors had left Africa between 50,000 and 70,000 years ago. That migration history gave those populations certain advantages in specific contexts: contact with diverse ecosystems, encounters with other human groups, exposure to new agricultural species. But it also meant that the story of humanity, as told by humanity, was told by people carrying a fraction of our species' total genetic memory. The populations holding the deepest archive of what it means to be human were largely absent from that narrative. Their history was not written down. It was written in their DNA. Researchers at the Smithsonian Institution's Human Origins program have noted that recentering our understanding of human prehistory around African genetic diversity does not diminish the achievements of any other population. It expands the frame. It reveals that the human story is longer, more complex, and more geographically rooted in Africa than the version most people encountered in school. The 300,000-year-old fossils of Homo sapiens, discovered at Jebel Irhoud in Morocco, described in a landmark 2017 paper in Nature, pushed back the origin of our species by 100,000 years and suggested that early modern humans were present across a much wider geographic range within Africa than previously known. The genomic data from the San and other ancient African lineages fits that picture perfectly. Our species did not emerge from a single point and spread outward in a clean, orderly sequence. It evolved across a continent in multiple interacting populations over a vast span of time. The San are the living evidence of present in communities across Botswana, Namibia, and South Africa today. It means that the full story of our species has not been lost. It means that the genetic archive assembled over 150,000 years of unbroken human presence in southern Africa is still available to science, still available to medicine, still available to anyone genuinely interested in understanding where we actually came from. The San did not survive this long because history forgot them. They survived because they were already home. Every other population on Earth descends from people who looked at the horizon and decided to walk toward it. The San are the people who understood, perhaps more deeply than anyone, that everything worth knowing was already here.